Hey class! Today we're learning about something really exciting. It's something that, as we talked about Mendel yesterday, something that, although Mendel ran some pretty awesome experiments, he never even knew about what we're talking about today, so it's really cool. We're going to build on some prior knowledge. We learned about mitosis a couple weeks ago, and then we learned about the basics of genetics and hereditary yesterday, and we're going to be building on both of those things to examine how our sex cells are produced. So everybody knows what sperm and an egg is, but now we're going to look at biologically, how do we make sperm and egg? So the course objective that we're looking at is to examine the process of specifically meiosis and relate it to the number of chromosomes, so production of daughter cells and gametes, and then looking at variation within a species. So to get started, the main similarity between mitosis and meiosis are that the phases are the same. The names are the same: interphase, prophase, metaphase, anaphase, except in meiosis, the cells go through the phases twice. So there are 2 rounds. So that's why you see prophase 1, metaphase 1, because we're going to go through again and there's going to be a second round. In interphase, which is just the time in between cell divisions, the DNA is copied. So if we look here, the dark blue can represent the father's DNA, or here, and the light blue can represent the mother's DNA. So we know that in a normal cell, humans are diploid, so that means we have 2 copies of all of our chromosomes, one from your dad, one from your mom, and we have 23 chromosomes, each with 2 copies. So 2 times 23 is 46 chromosomes. In interphase, they double, so instead of just having 46, you have 92. Instead of just having a version from your mom and a version from your dad, now you have a version from your mom copied, and then a version from your dad copied. So that's where you see this 4N over here. This word recombination, we're going to worry about later, so we're not going to worry about that right now. We're going to cover that in class and just work on a couple activities because that's kind of a hard concept. In prophase 1, we see that the chromosomes pair up, so the homologous chromosomes means that the version from your dad and the version from your mom that carry the same information but different versions of the same information. Like we talked about alleles, so you might have a different allele from your mom, a different allele from your dad, but the chromosomes are still aligned to the same. Those are what homologous chromosomes are. They pair up. They're very close together. So this X is 2 chromosomes, exact replica of itself from your dad, and then this chromosome is 2 chromosomes, exact replica of each other from your mom. But they together are homologous because they carry the same types of genetic information, just different versions. In metaphase, like we learned about in mitosis, the chromosomes line up on the metaphase plate, kind of in the equator of the cell. The microtubules are kind of playing tug of war with it, so they line up in the middle. We see our meiotic spindle has formed, and then in anaphase, the homologous chromosomes pull apart. So here in telophase, what you have is you still have your 2 copies from your dad and your 2 copies from your mom, but the copies are separate. So now instead of tetraploid, which is 4N, we go back to diploid, 2N, which means that there are again 46 chromosomes in each cell. And just a pointer, this diagram only has 1 pair of homologous chromosomes, but in actual cells you would have 23 pairs and they would all be lined up along the metaphase plate. So in this cell you might get 10 of your sister chromatids. This is called a sister chromatid, by the way. What that means is just that it is replicated but still connected. So this is the same genetic information on both sides. So this is sister chromatid. So you could have, like I said, you had 23 of these lined up on the metaphase plate. So you could get 10 from your mom, 13 from your dad, or just all different types of combinations. It's 50-50, it's random. We'll do a lab on that later, but just remember that in the end of telophase 1, you're back down to 46 chromosomes in each cell because the homologous chromosomes, your mom and your dad version, already separated during anaphase 1. Alright, the second round, similar but a little bit different. Prophase 1, or prophase 2, you have your two daughter cells, metaphase, they line up along the plate, and now instead of the homologous chromosome splitting, because they already split, the sister chromatid split. So I said a sister chromatid is just two copies of the same chromosome, so now those are splitting up, pulled apart. So now you go from 2N, which is diploid, to 1N, which is haploid. So meiosis produces 4 granddaughter cells, all with 23 chromosomes. Not 23 pairs, but only 23. If we go back, so we started out with 46 in a normal cell. They all replicated to make 92. Then your homologous chromosomes split in anaphase to put you back at 46. And then the sister chromatids split in anaphase 2. So your two copies split, giving you 4 granddaughter cells that are all genetically different, and that is the basics of meiosis. We're going to watch a quick animation, and there will be some questions during the animation for you to answer. Alright, so tomorrow in class we're going to be talking about more the difference between mitosis and meiosis, but for now, just a summary, the main difference between mitosis and meiosis is that mitosis is concerned with somatic cells, which are all the cells in your body that aren't sex cells. Mitosis produces exact replicas of cells, so if you have a skin cell, your skin cells are replicating all the time because you lose skin cells all throughout the day. So skin cells undergo mitosis to produce exact replicas of themselves, just to replace and replenish more cells. On the other hand, meiosis is for sex cells to produce gametes. So that's why in mitosis you start with 46 chromosomes, you end with 46 chromosomes. Whereas in meiosis, you start with 46 chromosomes, you end with 23. There must be that reduction in genetic information because in the production of gametes, you must reduce the genetic information so that when two different gametes come together, it forms a zygote with the correct amount of genetic information. So, big picture, what meiosis does is it reduces the genetic information of a person and makes that genetic information variable. So it's genetic diversity and reduction of genetic information into your sex cells so that when the sperm and egg unite, 23 chromosomes and 23 chromosomes come together to make 46. Because if you think about it, if every time a new generation was produced, we kept adding more and more chromosomes, we would have so many chromosomes. So that's why in meiosis the reduction of chromosomes is necessary.